Okay, without further ado, Dr. Zuckerman.
Yes. So again, thank you, Vivian, for the very nice introduction. And I'd like to thank everyone who's on the line today for tuning in for what George and I really think is a very important topic. It's a topic in applied clinical research where the role of the statistician is extremely important, and the need to work as a team of colleagues is extremely important. Next slide, please. Okay, and again, we'd like to thank the organizers, Michelle, Greg, and Vivian. Next slide. Our outline is the following. Number one, I will introduce the concept of patient-focused clinical thinking. There are diseases in cardiology right now that present an immense public health burden. And with our various standard approaches, the FDA has recognized that we aren't getting the biggest bang for the buck. And consequently, the so-called Breakthrough Devices Program has been initiated over the last few years, and I will explain how that operates. The good news about the Breakthrough Devices Program is that it allows for a very flexible clinical trials approach. This flexibility includes the possibility of using a two-stage approach, a seamless clinical trial approach, where initially a device may be approved based on intermediate endpoints. The trial may be continued to solidify the safety and effectiveness of the device with harder endpoints to get insurance approval. Now, this is a really important approach. Often the Bayesian power prior approach is used in this adaptive design. And Dr. George Chu will be focusing on some important work that he and his colleagues have done in this regard in part 3 of the presentation. Next slide, please. So we're going to be focusing first on unmet patient needs from the clinical perspective. Next slide, please. And we're going to use as an example today the area of heart failure. I think everyone probably recognizes that heart failure is a tremendous public health burden. One can see on this slide that unfortunately advanced heart failure, even in this day and age with some very potent drugs and devices, has a 5-year mortality for advanced heart failure that's very similar to advanced cancers. I can tell you that last week I was at the Heart Failure Society of America meetings, and I was sharing a panel discussion with Dr. Gibbons, who is presently the NIH's director of the National Heart, Lung, and Blood Institute. And certainly his point, along with many other people on this panel, was that there's a lot of room for innovation and new thinking in this area, given the burden that heart failure places on our society and on our patients. Next slide, please. So FDA Center for Devices has certainly been aware that over the last 15 years, medical device innovation has not always proceeded at the pace that is most beneficial for our society. And part of the reason has been certainly some regulatory challenges and constraints. And that's why starting a few years ago, the FDA Center for Devices developed the so-called Breakthrough Devices Program. It's become a significant aspect of medical devices development in the United States. It was specifically established for therapies intended to treat a life-threatening or irreversible debilitating disease where there are no available treatment alternatives. And for this audience, most importantly, new medical device clinical trial paradigms are being developed in response to the program and other aspects of our presently complex medical device ecosystem. And let me drill down a little bit more on that last bullet. The guidance is available through the FDA, and one of the key points it makes is that initial approval for a breakthrough device may be made on the basis of intermediate endpoints. And let's go to the next slide so that we can dive a little bit deeper into CDRH use of intermediate endpoints. So the way we define intermediate endpoints is that it measures a clinical or physiological outcome that can be assessed earlier than an effect on irreversible morbidity or mortality, and that is considered reasonably likely to predict the device's effect on irreversible morbidity or mortality, or the intermediate endpoint in and of itself is a useful measure of clinical benefit to a patient. So let's look at some examples in the cardiovascular arena. They would include angina frequency, quality of life, exercise function, brain natriuretic peptide, which is a good marker of extent of heart failure. So I think we can conclude that most intermediate endpoints do not fully meet the Prentice or Hughes surrogacy criteria, that is, true substitute endpoints, nor do they have to. So how do we put this all together? Well, we can use a collection of intermediate endpoints at the initial stage of device approval to really obtain safety and effectiveness evidence that we are improving the pathophysiology of the disease. And let's go to the next slide. Now, again, the criteria for the breakthrough program are criteria 1, we provide for more effective treatment or diagnosis of life-threatening or irreversibly debilitating human disease or conditions. And we have one of the following subparts from criteria 2. So the bottom line is that these are devices that are intended to improve important aspects of our present healthcare system. Next slide, please. So let's put it all together and let's use a heart failure device example. One approach is to simultaneously meet several non-mortality heart failure hospitalization endpoints. For example, in a heart failure device trial, we use a combination of several key intermediate endpoints at the first stage of approval. This could be 6-minute walk time, a measure of exercise, KCCQ, or what's known as the Kansas City Cardiomyopathy Questionnaire, which is a good marker of how the patient may be improving in terms of their symptomatology of heart failure. And then often a third endpoint, such as a biomarker called BNP, or brain natriuretic peptide, which allows us to again see that the heart failure is improving. And we look at the totality of the data, and the final analyses should support a reasonable physiological mechanism and a clinical benefit. And I have put one reference at the bottom. Next slide, please. This isn't to say that the FDA Center for Devices Cardiovascular section ignores mortality and morbidity at stage 1. In fact, we usually insist upon some type of predictive modeling at an extended time point for morbidity and mortality data to further strengthen conclusions about the benefit risk assessment. For example, we might try to predict what would occur at 2 years, given some of the limited data that we have when all patients are followed out to 6 months. And a good reference is again provided on the bottom of the slide. So let's go to the next slide. So here in cartoon form, we have the makings of a seamless clinical trial design. And I know this slide is a bit complex, but there are several important aspects that one should hone in on, and George in his talk will be honing further in on. The first aspect is that after 6 months, after 6 months of data are collected on the 3 intermediate endpoints, and we also have data on mortality and hospitalizations, we can make an initial submission to the FDA. In this submission, we expect that all 3 intermediate endpoints will meet their pre-specified hypotheses, A, and B, that using predictive modeling, that the trend for heart failure hospitalizations and mortality at a later time point will be what we're expecting. So while we have that submission in-house, the sponsor can continue to accrue patients. And this is important because while FDA reviewers are working on the first submission to possibly give the sponsor a label for improvement in patient symptoms and physiology, they're accruing additional patients with an adaptive design. And hopefully at the end of the day, they are able to stop accruing patients and reach a conclusion later on that there's an improvement in heart failure hospitalizations and mortality. Because certainly this is another important consequence of an effective heart failure treatment. And frankly, the insurers are looking for this type of data before usually they will reimburse a company for a new device treatment. Next slide, please. So this two-stage seamless trial design has caught on in the heart failure device arena. In Vivian's excellent introduction, she indicated that most device companies are unable to do very large randomized trials, certainly with their first pivotal trial. And this trial provides many advantages by being broken up into several parts, but yet have a seamless, continuous aspect. And the references here, again, demonstrate that many heart failure device manufacturers have taken up this theme. Next slide, please. So at this point, I'm going to hand the baton over to George, who's going to develop these concepts and approach in more detail so that we can all appreciate some of the important statistical and clinical reasoning behind one paradigm for the Breakthrough Devices program. George?
Oh, thank you, Bram, for nice clinical sharing about this urgent need to promote public health through the Breakthrough Device program. So in line with your clinical perspective, I'm going to present recent innovative statistical methodology, how to leverage external data, but in the same time, maintain the rigorous requirement from regular perspective, so-called reasonable assurance of device safety and effectiveness.
You're on mute now, George.
Oh, I don't know why I got unmuted. Okay, stay. Can you hear me now? Okay.
Yes.
So first, I'd like to make a note. Thanks to my co-author for the paper I'm going to share. Laura Thompson is the leading author, and other co-authors, some of them already left FDA. I'd like to thank my current boss, John Lessinger at Edwards, to support my time for this webinar. And also, our paper actually is kind of motivated by Tariq Haddad paper 2017.
Somehow you got muted again, George.
Okay, next slide, please. Yeah, so as Bram already illustrated, that we have a device breakthrough program and kind of two-stage seamless design, building in some adaptive feature with the interim analysis at 6 months for all the patients enrolled reach 6 months initial sample size. And the reason for doing that, because oftentimes device company cannot commit a large big capital upfront. So for a large trial, like a drug trial, you have like 2,000, 5,000, but usually typically for drug trial, we're looking at most like 800 up to 1,000 patients. So initial sample size, we're shooting for the soft endpoint, like heart failure study, biomarker BNP, KCCQ, quality of life, 6 minutes walking distance. And we the 6 months follow-up data, potentially you get a statistical win on so-called intermediate endpoint as listed here. But in the same time, we want to have certain confidence that hard endpoint in terms of the CV deaths, hospitalization due to heart failure events will trend in favor of the device as compared to the randomized control. But oftentimes at this time, we have a smaller sample size, short time follow-up. We cannot secure statistical significance for the hard endpoint. So we basically try to build a called Bayesian predictive probability to project how likely to the end of the whole trial with a larger sample size, longer follow-up, what's the confidence we're going to have a statistical win on the CV deaths and HF event by utilizing the correlation between the intermediate endpoint and the hard endpoint. Because at that time point, some of the patients have longer follow-up, probably already have such event. Some of the patient does not have. So we based on the correlation structure built in the predictive probability, get some feeling about it. So to augment that Bayesian calculation posterior probability, if you have a good external data, that's where you potentially could augment the posterior probability to have higher level of confidence decision making to continue the trial or just stop there for fertility. So I will illustrate one potential methodology to leverage external evidence based on Bayesian power prime. I'm sorry, I have to ask the host to move the slide. I always try to move stuff by myself. That's why I got muted again. Okay, so the reason is that device trial often more expensive than drug trial on the per patient basis. Usually, especially for cardiovascular device, the surgeon cost implant, we're talking about 25,000 to 50,000 per patient. So that's why it's really important for us to rely on some good external data if that's possible and acceptable. And so there's a need for efficient, flexible design, especially for heart failure patient, which we have high unmet medical need. So how to do that? Oftentimes for medical device, we don't have the luxury have a lot of external relevant good quality data. And that's why in this talk, we only focus on only one external data available. And other potential in the future, external data available for device, if it's well understood device area, they have so-called virtual patient based on the simulated data, based on the engineering bench and previous clinical study. For example, recently there's so-called Living Heart project. The Hadas paper actually is driven by that project, leveraging virtual patient data to augment real patient data in the pivotal. Next slide. Recently, the MDIC, is a medical device in consortium, published a kind of white paper about the framework external evidence leveraging. So if you go there, you will find a variety of statistical methods with some case example, a real submission example. The methodology is mainly like Bayesian hierarchical model or meta-analytical power approach if you have multiple external data set available. But in this talk, we just limit our scope on the single external data set built on Hadas methodology with discounting function dynamic borrowing. Next slide, please. So overall, to simply put that, it's maintain reasonable assurance, not only based on the limited current smaller trial, we need some prior relevant high quality data. So we combine them together.
我说你可以开了吗？
Could you mute your microphone? I'm
上
sorry.
错了。
Okay, next slide, please. Next slide, please.
可以就下去一个抽口下去，接着上来。
So the paper I'm going to
我不想
focus
开，
on is highlighted in
我对
yellow
开
in
没
this
什么兴
slide.
趣。
But recognize there's some other methodology already published available. And the first one is so-called the meta-analytical predictive power using the conventional power prior, just a non-informative mixed with informative, kind of discounting through this mixture. And Haddad's paper built on the virtual patient idea, and we extend that to accommodate some concerns associated with the whole data set to determine the power parameter. So we have the Laura Thompson paper just recently published. And also one paper is based on the propensity score adjusted power approach. Actually, it's more than one paper based on that propensity score integrated approach. Next slide, please. So here is just a motivating example for you to have better understanding the following key points. So suppose only single historical data available to be borrowed, and D0 is a prior data for the investigational device. It could be from the early feasibility study, or maybe a first generation device, and could serve as a historical control arm, or even just augment the device treatment arm. Hypothetical single arm study for new orthopedic device. This example is actually detailed in the published paper. So it's a continuous variable, ODI score improvement from baseline. We're looking at the 20 point, succeed or not. And historical data could be like 300 patient data. So we want to make sure kind of borrow information from D0 without over inference from the D0 to the overall posterior probability to draw the inference. So one attractive option is Bayesian power prime. Next slide, please. So here is very kind of review of the Bayesian power prime, very simple. The power prime is using the so-called alpha zero as a like discounting. That's the way to achieve the discounting. If the alpha zero is fixed somewhere between 0 and 1, I would determine the percentage of borrowing from the prior data. So you have the L0 is the likelihood of the prior data, and pi theta is a kind of non-informative prior. From these two, you derive the power prior, pi pp. And posterior basically plug in the pi pp as kind of another prior in combination
你可以
with
从
the
这儿
new
出
data.
去，你再转回来。
I'm sorry, somebody please mute your phone or microphone. So the new data is the theta, it's a current data set, it's a likelihood times the power prior. Then give you the posterior. So it's kind of prior, kind of weighted average from the two parts, the current data likelihood plus the power prior. So how to decide alpha zero? It could be fixed upfront. Usually for our device trial, based on the clinical thinking, we come up with the maximum to be borrowed. And so the approach is how to determine fine-tuning the degree between the 0 and the maximum. So next slide, please. So mainly it's a clinical decision, but also usually for Bayesian adaptive design, using such approach, we require sponsor to do extensive simulation to ensure the frequentist property, operation property like type 1 error is well controlled at acceptable level. Inevitably, the type 1 error will be a little bit inflated to maybe more than 5%. But as long as we accept level that's a kind of one factor to drive the calculation of the alpha max. But first of all, we have to make sure the external data is clinically relevant, fit for use. So based on the clinical understanding of the target patient population and any major change to the device design, and basic idea, we have to avoid any over inference from external data. Next slide, please. So let me just briefly review Haddad's paper. It's called a Dynamic Power Supply Approach, DPP. This alpha zero power parameter will be depends on kind of similarity using hypothesis testing to derive the p-value between D0 and D. D0 is a prior data, D is the whole current data set in terms of the clinical outcome of your interest. And it's capped by, as I said, pre-specified Rmax driven by clinical thinking and simulation to ensure the type of error at acceptable level. And then they also plug in so-called Weibull function to fine-tuning the degree of borrowing based on the hypothesis testing between the D0 and the whole data set D. So there's two improvements from that Haddad's methodology because some concerns associated with using the full data set of the current data. So we built in kind of interim analysis to further downgrade the degree of borrowing at the interim. So you're not going to use the full current data set. And also, instead of the hypothesis testing derive the p-value for the difference to judge the similarity using Weibull function to fine-tune the degree of borrowing, we actually plug in a so-called equivalence margin driven by clinical reasoning. So this margin should be small enough to be considered as clinically irrelevant difference. And we also force the observed mean between the current and the prior will be fall into the similarity measure. That's a key feature to further control the type of error and the bias. Next slide, please. So just brief, alpha max pre-specified with clinical consideration, simulation to control the type of error at acceptable level with desirable power. Next, please. So the interim actually give you also opportunity to potentially increase the sample size if your similarity is less than you expected and the borrow is far below the maximum up front. So at that time you could have the opportunity to expand the sample size for the second stage, D2 here. So compared to Haddad's methodology, instead of using both the whole data set D1 plus D2, we only use interim D1 data to fix the alpha zero. And the similarity measure, instead of the difference less than zero, we plug in the equivalence margin called delta here, given day 0 and day 1. And also we force the restriction, the observed difference have to be fall in within the weighting minus delta to delta. Next slide, please. So basically, the advantage of this introducing such a similarity measure is try to more tightly control the bias and type of error. And also when everything is prefixed at the design stage, the magnitude of the delta, the alpha max, these two values have to be fixed through a thorough discussion with the clinical team on the both sides, sponsor and FDA, have to be agreed upon. Next slide, please. So here's a very simple illustration for details and simulation. Please refer to the published paper. And the idea is with such a equivalence margin, we try to avoid a scenario when the sample size is smaller, you potentially borrow more. So even though they could be far away, but because of the small sample size, you have a more spread distribution, you still end up borrow more. So that's the drawback with the Hadass decision making without the delta equivalence margin. Basically, it's a test for the difference, whether or not it's significantly different from 0. So the p-value around 0.5 is kind of identical, then it slide upward and downward with the Weibull function to determine the degree of borrowing. Next slide, please. For our methodology, because of the delta introduction, we only limit the borrowing when the observed mean difference is within the region, and also the posterior probability of the whole region less than delta, between minus delta and delta, will be times to the alpha max. So the final degree of borrowing will be somewhere between 0 and alpha max, depending on the posterior probability of such similarity fall within the delta. So with this way, we actually borrow more when the sample size n is larger, effectively avoid domination by D0. So that's why for our approach, we don't want to have a too small percentage of D1 to determine the alpha 0. So at the interim, usually we recommend about 50% to fix the alpha 0 at the interim analysis. Next slide, please. So in summary, our conditional power prior approach for dynamic borrowing is based on the interim analysis and Bayesian posterior probability of optimum similarity within a pre-specified clinically acceptable margin. Then degree of borrowing is further fine-tuned by the posterior probability of these two data sets in concordance with each other. So that will avoid any over-inference from external data set. And the introduction of the interim analysis could further ensure the power as needed if the borrowing cannot be as much as you need, as you expected. And also, bias could be lower and the type of error well controlled and acceptable level. Next, please. So in relevance to the propensity score methodology, that approach, they call it outcome-free approach. Basically, that approach, the alpha max is fixed. So upfront, the degree of the borrowing for propensity score methodology is fixed upfront. That cannot be changed. The fine-tuning only applicable to the different stratum of the propensity score. So basically, they allocate the maximum borrowing differentially according to the similarity within each propensity stratum. But that approach actually will not control the alpha max, cannot fine-tune alpha max. So sometimes could still be fall into the problem over inference from the external data set. But actually in real world practice, when sponsors select external data to be borrowed, usually it's already know that external data is in favor of the alternative hypothesis. So the weight maybe when we determine the alpha max, maybe we can use a propensity method to come up with alpha max in combination with the clinical thinking, and then use our dynamic methodology to have the interim analysis to confirm or fine tuning down the alpha max. Maybe that's another way for the future. Next slide, please. So here's a couple of reference. Our paper is published 2021. So hopefully you have time to read the paper. If you have any more questions, just reach out to Laura and myself. We will be able to address any question you may have. Next slide, please. Okay, so I turn over to our discussion facilitator, Greg.
Thanks, George. And thanks, Bram. That was a very interesting and motivating example of the partnerships between clinical and statistical scientists. And that's exactly what this webinar series is about. And it's particularly exciting to see such a sophisticated methodology that's developed. So that before I get into the first question here, I want to remind people that you can enter questions into the chat. And we're going to do this interactively. So if you have a question, you can be part of the discussion, but we're going to use the chat to kind of organize the questions. There's already a few in there, but go ahead and enter some more. I get to ask the first question, but when your question comes up, you can unmute yourself and talk with George and Bram. But when you're not involved in the discussion, please mute yourself so we have fewer distractions. And I also want to take this opportunity to apologize for the technical difficulties we had a few weeks ago. And I'm very glad to see that all or most of everybody came back. We have a very good showing here, but clearly George and Bram have generated a lot of interest. So the first question, recognizing that statisticians are often uncomfortable dealing with non-quantitative information, analyzing non-quantitative information, and recognizing that clinical professionals are often uncomfortable with more sophisticated statistical methodology here. You each bring some very advanced and sophisticated ideas from your areas of expertise. How have you been able to reach across the aisle to engage with each other and collaborate with each other so effectively to produce such a powerful and useful and integrated approach?
Greg, can you hear me?
Yes.
Okay, so I think this is a key question. And the first thing I think we've recognized at the FDA is that one size doesn't fit all. More standard approaches aren't getting us where we need to be a lot of times, especially in the areas where we need to innovate. There's great potential, but the ability to do what I would call applied clinical research has not kept up with some of the basic science. So I think you've put it very nicely. We have to get over some of our prior boundaries and work together as a team. And we've been lucky at FDA Center for Devices because for a long period of time, Dr. Greg Campbell was our chief of Biostatistics, and he was very effective at training medical officers to appreciate Bayesian statistics, to think more quantitatively. I think that's part of it, training. And with our statistical colleagues, we need to communicate more effectively so that they can at least appreciate the so-called art of medicine, or the qualitative approach that's often necessary to at least initiate a problem. So that's a long-winded answer, but I think the real essence is what you indicated. Many times the current system is not going to provide you with what we need. So we have to change the system, be able to work together as professionals and just get on with it.
Very, very good point. You know, there's especially certain areas, there's a great need for something new and innovative. You can't do it with the standard approaches like you demonstrated here. And so hopefully that's motivating to people, right? Because you have to be uncomfortable a little bit and reach across the aisle, go a little bit further across that bridge where you can meet in the middle and work together. But I think you also brought up a really good point, using Greg Campbell as an example. Need champions like him who are very effective at this, you know, as a statistician, communicating at least conceptually with clinical people, and similarly finding clinical people that are very effective at understanding the statistical concepts and communicating the clinical ideas to statisticians in a way that they can make better use out of it. George, you want to provide your perspective on this question?
Oh, yeah, exactly. As Bram and you very nicely stated, I just like to add on, it's really driven by patient need. So I really like the current, everybody think like a patient-centered, patient-focused approach. Because for breakthrough device program, it's really to address that high unmet medical need. So in our regular setting, we call it promote public health. So that's a part I really like it. So to promote those breakthrough device to meet the clinical need, we really need some unconventional methodology to have a more efficient and flexible study design. But in the same time, we still have to hold ourselves to higher standard to reach reasonable assurance. So that's kind of balance we're trying to achieve.
Very good. And again, you guys are a motivating example. I think our audience appreciates the very concrete example you provide. So going to the questions from the audience, the first, well, actually first two are from Terry Johnson. So Terry, if you want to unmute yourself and get ready to be part of the discussion. The first question here for Bram, what is the difference between PMA approval versus PAMS approval in terms of being able to market the device, timing to market?
Okay, so that's a great question. And just by way of background, I had the privilege of working with Terry for a number of years while she was at FDA, and she's asked a great regulatory question. So with the initial PMA approval, we would indicate the device, the hypothetical device, to be used in heart failure in order to improve symptomatology and exercise duration. With the subsequent PMA supplement, hopefully we could add also decreases heart failure, hospitalizations, and mortality. So after the first approval, the labeling is more restrictive, which puts the onus on the manufacturer to not advertise the device for something other than its indicated label. But by the same token, we're usually working with small companies with limited capital, and it allows them to gain more venture funding and other resources to keep going. So this has been very helpful for the device startups. With the more expanded label, what the manufacturer is intending is to get a better ranking in the heart failure guidelines, and also to better ensure insurance coverage. For example, if one only stops at improvement in symptoms or part 1, probably the guidelines would be a class 2A at best. But if you can show a reduction in mortality and heart failure hospitalizations, then you're going to get a class 1 guideline recommendation, which one is very helpful for treatment of heart failure patients, and 2, sells a lot more devices, frankly.
Terry, do you want to clarify anything in that question, or should we move on to your second question? Well, let's move on to the second question. This is for George. Is there some criteria for selecting the endpoints for phase 1? For example, they need to have certain level of independence, correlation to the endpoints for phase 2. Do we need to consider all these endpoints to control for type 1 error?
Yes, so this is a very good question. To my understanding, we do expect the intermediate endpoint will have a good correlation with the hard endpoint. That's why we try to make advantage of that correlation to do the predictive probability calculation for at the interim. And by the way, to my understanding,
I don't know. What do you need?
Meng, please put yourself on mute. Thank you.
So usually for our current breakthrough device, at the interim, in order for sponsor to make early claim for the soft endpoint, usually we look at the totality of all the soft endpoint have to be met based on the design stage. So like biomarker significantly decreased in terms of BNP, or in the same time, we see a significant improvement in both KCCQ or 6 minutes walk without any harm or trend in terms of the heart failure and hospitalization event. So that way is very much controlled. But overall, the simulation actually give us the idea how much inflation type of error will occur It will be somewhere between 5 and to acceptable level, maybe 7.5, or maybe even sometimes up to 10%.
Yeah, and that's going to be very important because for regulatory decision making, you need to have a lot of confidence that the surrogate is correlated with the clinical outcome.
Exactly, yeah.
So let's take a step back, Greg. You and George have had a good discussion, but Terry's second question is a very good one in that it points out the ClinStat discussion on choice of endpoints is an important one between sponsor and FDA review team. Primarily, what we're looking for with the intermediate endpoints are, maybe 3 endpoints that, when put together, give us a good level of reasonable certainty that we're making an improvement in the pathophysiology of this disease. When you look at the example, for example, exercise time, the patient-reported outcome, and the biomarker brain natriuretic peptide aren't among themselves that well correlated. Maybe the R value is 0.30 or something like that, correlation coefficient. But when put together, it's because of that, can be a reasonably persuasive package that you're truly doing something positive. So these endpoints are reasonably independent of each other, but it really takes some thinking for each specific area. That this hypothetical example was for devices for chronic heart failure. There's also a big push right now to develop devices for acute decompensated heart failure, where the choice of endpoints is a really interesting one.
Yes, that brings up another idea, I think, that we need to think more algorithmically rather than, being driven by the statistics, because that's a way to bring both sides together in coming up with a way of understanding the endpoint, to define the endpoint in such a way that it's correlated with the ultimate outcome of these clinical events. And that's more of a new way of thinking, right? It's these, how do we work together? And this would be a great example of that.
Right.
So moving on, Roseanne has a question here. Can the methods outlined in this talk be applied to de novo 510K? I can't explain that question. So hopefully you understand it, or Roseanne can chime in here.
Yeah. What Roseanne is referring to is, in addition to the so-called PMA or pre-market approval example that we gave, there are other pathways for approval depending on degree of risk and similarity to prior devices. One pathway is the 510K pathway. Another pathway is the de novo pathway. And the general response I would give to Roseanne is that if you're dealing with a device that requires clinical data, first we need to figure out what are the essential questions. And if you think the design can be more efficient using a particular Bayesian design, then go for it. So there's no hindrance.
Good. A couple people were asking if they can have the slides afterwards.
Absolutely.
Great. And a very positive comment here. Thanks for an interesting and stimulating session. I have another question I'd like to ask. The validity of external clinical data is essential, especially when it is intended to support regulatory decisions. In practice, data with positive results, trends, are more likely to be selected. How to evaluate the potential for selection bias here?
George, do you want to go first, or would you like me to go first?
Yes. Yes. We statisticians always love the topic of bias. So it's really true. I mean, for any multiple data sources out there, which one to be selected to be borrowed, obviously there's a potential for selection bias. And so the first protection line, I think, have to be the sponsor have to talk with FDA, how they come up with the selection of particular data set to be used. Okay, so usually I think it's a conversation between medical officers and sponsor. From statistical point of view, we probably need to, if it's multiple data set available, there's an approach called a meta-analytic prior, or maybe Bayesian hierarchical model could address that potential bias there. But for most of the device trial, usually it's a very relevant high quality data is very rare as compared to a drug trial, which tend to have more data set available. So for device trial, I would think the power prime to have a further discussion on the alpha max it is a way to address that potential random high bias.
Okay, and to add on to that, I would underline a few things that George said. Number one, we'd like early interaction with the sponsor to discuss a potential Bayesian design. It's really critical. It takes more interaction up front. When looking at a possible data source, and it's common that the data source will be from outside US, specifically Europe, I think in addition to what George said, the medical officer, the statistician needs to be involved to look at similarity of definitions, procedure, protocol, clinical events committee adjudication, etc., similarity of devices used, and so forth. But be it as it may, we're always concerned with the possibility of bias. And I think then it goes to, simulation of the actual trial and what is the maximum amount of prior data that could be borrowed from external data sets. And I can tell you empirically, the FDA Center for Devices Cardiovascular Division has settled on a value of about 30% borrowing, A. B, in terms of the type 1 error control, we don't like to see situations where the alpha goes above 0.10. And then at the end of the day, and this is shown in the cardiac calcium modulation device reference for anyone interested, it's interesting sometimes to do in a post hoc fashion a analysis to see, sure, your trial was significant, say with a predetermined 25% borrowing, but how much did you really need to borrow? Was it 25%, 20%, 15, 10? You can do that sort of sensitivity analysis to see at the end of the day how important borrowing was, and that may change your perspective a bit if you could have borrowed less. So as George mentioned, it's an important problem and we need to think about it at multiple steps, Greg.
Very good explanation. And I'd like to bring up another analogy here. If you think about doing a meta-analysis, a lot of times the discussion is focused on the statistical methodology to avoid Simpson's paradox, right? So there's statistical methodology you can use to make sure that you get an accurate inference from the data. But what is often forgotten about at that stage is there's a lot of clinical considerations, right? What should you be pooling these studies together, right? If they're really different populations, what does it mean if you analyze them together, right? The result is going to depend on the proportion of the two populations in the meta-analysis. So again, this is an opportunity to remind ourselves that we need to think about it from both sides, the quantitative side and the subject matter expert side. And it's not something that we need to handle with just the statistics, because if you set it up wrong, you're not going to get useful inference from your analyses.
That is a great point, Greg.
Thank you.
All right, it looks like we have time for probably one more question here. So the similarity measure based on pre-specified clinically appropriate equivalence margin is used to determine the degree of external borrowing. Could you please elaborate on the general considerations for the selection of the equivalent margin?
George, do you want to go first or should I go first?
I think this is mainly driven by clinical reasoning with some implication on the statistical properties. So I like Bram go first, and then I will talk about the implication of the delta on the operative properties of the borrowing.
Okay. So George, be glad to go first, but I think all these questions are really ClinStat questions. We can't separate the two because, initially, let's recognize that one size doesn't fit all for the delta margin. So it really is going to be dependent on the device and the endpoint chosen. And the clinician can certainly give a starting guesstimate of what the delta should be. But I think this is where we're going to need some simulation to see with a certain range of sample sizes, does this leave us in a situation where we can also balance some of the other issues that we were just talking about. Certainly, my main one response would be, well, the delta should be as small as possible. But, if you take it to that extreme, we aren't going to have any trials of this type. So we have to get a reasonable compromise. And I think that could be shown with the sponsor doing a number of simulations with the delta being smaller, larger, around the delta that the sponsor actually has honed in on and thinks is appropriate. George, would you like to take over?
Yeah, Bram, you already stated the implication very well through sensitivity. The reason, the smaller the delta, the less likely you're going to borrow, because our methodology force the difference within the delta. So if it's smaller, the posterior probability will be smaller. And then you have very, very limited power, probably approaching to zero. So you do have to kind of balance through the sensitivity simulation. But obviously, first driven by clinical thing, you have to be small enough. You feel comfortable from clinical perspective, is irrelevant. They're similar from clinical. And then how small to go, then you're probably limited by the simulation to see whether or not we still can control the type of error below 10%, but in the same time, ensure enough borrowing so you still have a reasonable sized pivotal study to be feasible to be done.
I think something that really shines out here from your answers in your conversation is how much you recognize the value of the other discipline, and you keep bringing each other into the conversation. And you both used similar words, talking about the compromise and the balance. And I think that's what it's all about, is these two disciplines working together in balance. But we actually have time for one more question. So to communicate with each other, Bayesian methodology is, I think, very well suited for this cross-disciplinary scientific engagement. And a couple things in particular, I think, are very useful for creating that bridge and finding common understanding. For example, the Bayesian probabilities, I think, are very easy for everybody to talk about, to kind of quantitative the level of evidence. And also simulations take very complex ideas, and then you can see what would actually happen, make it more concrete. So I don't know if with the last 5 or so minutes here, maybe you can talk about these tools and how they've aided your communication with each other.
Bram, you go first.
Okay, so I think you're right, Greg, but I think at this point, many clinical trialists in the community still fear Bayesian approaches. And I think there's a real need in the cardiovascular literature still to write articles that present the key aspects of Bayesian statistics, like you just enumerated, in a graphical approach, such that communication can continue to be increased. So the bottom line is, I think you have developed a good idea for a journal article.
Yes, really promoting Bayesian analyses in a space where it's very relevant and useful in a learning and decision-making environment like this. George, you want to finish up?
Yes. Yeah, I think I just realized my former boss, Ram, is in this audience. So Ram is a division director, served us very well for the past several years before he moved to the current BMS company and methodology head there. So Ram, maybe you're a Bayesian expert, and maybe you can provide us your insights for your experience in Bayesian and Bayesian application at CDRH.
Another great champion like Greg Campbell. Yeah, Ram, please.
So Greg,
provide your
thank you.
perspective.
Greg, thank you. And also thanks to George and Bram for excellent presentation. And well, first of all, CDRH is champion for using the Bayesian methods anyway. So that's pretty known. And Greg, Campbell has already established that, and there is a guidance. And now CDR is also kind of getting there, but still there is a lot of reluctance, and more so because the drug is not equal to device. Drug is a very different thing. That's what you hear every day. And also, clinicians there are not like Bram, open to new methods and new ideas. They seem to be still kind of fixed on using the frequentist methods. And for them, unfortunately, the p-value is the posterior probability. So having said that, George, I just wanted to ask a question to you and Bram, even about the choice of this alpha that you talked about. It depends on the outcome at an interim analysis. And this interim analysis is not performed for futility or efficacy, and this is not based on any information fraction using alpha distribution, type 1 error control. This is just to get a interim data D1, so we can compare the D0 estimates based on the D0 and D1 and adjust the alpha in some way that comes from only from propensity score. So that's a very clever idea, good work. But I am asking Bram, in this case, how do you suggest as a regulator, if we are not going to do interim analysis, if we are not planning for that, how do we come to you to say that I want to get a interim data D1 to refine my estimate of borrowing parameter alpha, and that I may have just based on the propensity score, which is only looking into the similarity of the baseline covariates and so on. Is that acceptable to you and CDRH, that you will allow us to come up with some kind of suggestion to collect a small part of the data D1 at interim?
So George, would you like to go first, or would you like me to go first?
You go first.
Okay. So Ram, I think your general comments are very good before we get into the specific question. Sometimes it can be difficult to change the path of an organization like FDA. I would ask that people, though, not give up, because as George has mentioned many times, ultimately, we're all interested in improving patient care. Now, Ram, you've put, I think, the main issue before us on the table. This is a new approach. When you were division director, you really helped us move the power prior approach forward, and I hope we can continue. And so in a situation like this, if you, came in with a device to the cardiovascular division, I would suggest that in addition to the regular review team, you might consider requesting that I attend the meeting, and I would be happy to. Or, as you know, sometimes sponsors meet with the review team and they don't believe that there's been effective communication between the sponsor and the review team. In that case, the sponsor should never hesitate to call an office director like myself with their concerns. Frankly, it's part of my job. I spend about 10 to 15% of my time talking with sponsors with this type of issue, since we're often on the cutting edge of technology and we try to rectify things. But the bottom line is that we would encourage you to challenge us with these sorts of ideas. Does that help you, Ram?
Yeah. Thank you, Bram. Thank you so much. And George, I need to hear your final remark on my question. The choice of alpha. The choice of alpha depending on interim data.
For our current paradigm, the example we talk about, actually, basically, you could think of the first stage as an embedded, separate trial, only shooting for the soft endpoint. So the alpha, we're not distributing the total alpha in the two parts. We just, kind of embedded trial within the big trial, which is shooting for the hard endpoint. So I don't quite understand. We need to control alpha, kind of allocate the different alpha to the different parts.
That's good. So again, thank you, George. And Greg, I don't have any further comment, but just wanted to mention that this was good to see both Bram and George, and you also here. And I am fortunate enough to have co-authored the paper with George, as well as with other FDA colleagues when I was there, and I still continue to interact with them. So thanks, Bram, for the opportunity I had while I was there.
And
I'm so
similarly, it was
glad-
really nice to hear your perspective. I think you added a lot to the conversation.
Absolutely.
Yeah, I'm so glad to see so many familiar names on the audience. I see Roseanne White, Terry Johnson, all my former colleague or same field, medical device. I'm not sure how many audience from medical device industry, how many of them from the drug industry. Is there any way we can get an idea?
We don't have a way of polling, but that's a good idea. If we, assuming we continue this next year, to maybe add that interactive component to it, that would be a good idea. Now we're over time here, so I'd just like to remind people that we have one more webinar in this series. It'll be on October 26th. I think the advertisement is already out there. It'll be from 12 to 1:30. The title of the webinar is Cardiovascular Adverse Events in Oncology Trials, Understanding and Appreciating the Differences Between Clinical Trial Data and Real World Reports. And the two presenters are going to be Jay Herson from Johns Hopkins and Michael Ewer from University of Texas MD Anderson Cancer Center. So hope to see you there.
Thank
Vivian,
you.
did you want to say anything before we close out?
Oh, I just want to thank everybody, all the speakers, and thank the moderator and all the participants. This is really an eye-opening seminar. It was great. Thank you so much, and I hope we'll see you again on October 26th. Again, collaboration between clinician and statistician. Thank you.
Have a good day, everybody.
Yes.
Bye.
Thank you all. Bye-bye.
Bye.